how you got into this work, what's new and shiny in your laboratory, and how, and sort of some impressions, you have about how to move the field forward. And we can go through those sequentially and everything, but I'd love to just, hear what you're, everything about what we talked about. Okay.
So my major interest is actually in kidneys. Okay. So and that's actually where I started. I started in paediatric nephrology at the Royal Children's Hospital in Melbourne. Mm-hmm. And I was particularly interested in how when oxygen was not supplied to kidneys, which happens under lots of situations, you actually have problems later where your kidneys give up. But what that led me to was doing a PhD in that area. And then I had a very famous researcher in diabetes complications in kidney disease see me at a conference present. And he said to me, you need to work in the diabetic kidney. You need to come and work with me. And so I did just that. And that was the start of my association with JDRF, actually. So that was my first postdoctoral fellowship. And I've had a fellowship, an advanced postdoctoral fellowship, a career development award. I've had many other things in my complications field with them. And the beauty of that stuff is that it actually allowed me to develop this area of advanced glycation that I'm interested in, because probably what you don't know is that that chemistry that I'm interested in has been associated with diabetes for a very long time. And so hemoglobin A1C that you measure in patients to look at their glucose control, that is actually part of the chemistry. So it's an intermediate of advanced glycation. Okay. It's where sugar is bound onto hemoglobin. And it's like a litmus test for how patients' glucose has been going over a longer period of time. Right. And actually, advanced glycation is the next step of that, where it becomes a permanent addition of the glucose onto the sugar. So hemoglobin A1C can go back and forth. It sort of accumulates, and then the blood cells are turned over, so it disappears from the body and starts again.
3 months,
yeah, exactly. That's right. So that kind of gives us about a 3-month window, but advanced glycation kind of gives us a bit of a longer window. And so I did a lot of work in bringing some therapies through for diabetic kidney disease, and we had one come through into phase 2B clinical trials in that space. But it happened to be in the global financial crisis. And so the company that had the compound died, unfortunately, and so did the trial. There was our trial in diabetic kidney disease, in type 1 diabetes, and there was also a heart failure one going at the same time. And so I sort of had to step back at that point, because obviously, that's a big blow when you've taken something through to that late stage. But one of the pathways that we were also interested in as we were going along, so I changed tack a little bit and started to look a little bit in type 2 diabetes. And what I found then actually was that these advanced glycation inhibitors, and looking at RAGE, there started to be some modulation of glucose control. And I hadn't seen that before in my type 1 models with complications, because normally we would take away the islet function, and then these models will develop kidney disease, cardiovascular disease, and those complications. Yeah. So we sort of were missing that effect of the drug. And so that started me thinking. And so I did some studies where I guess what we did was strip it all the way back and said, okay, well, if we just give advanced glycation end products, so you can actually eat them in the diet. So there's something that food chemists use to impart particular properties to our food. So things like, and they're desirable properties. So they're things like longer shelf life, aroma, taste. So in its most simplistic form, it's that brown colouring, so the food that's yellow brown that we love and gives us that beautiful kind of reward feedback.
Doritos.
That has Doritos, bakery food, cereal, all of that is a very high AGE kind of content food. And certainly we know that there's been an escalation in what we've been taking in, perhaps since about the 70s, sort of since food processing became the more norm. And so I wondered,
I teach nutrition, so I'm following you.
Looked there, but much to my surprise, when we fed these AGs for a very long time, we didn't just get kidney damage. What we actually got before that was actually some changes in the islets. And not just changes in islet function, but actually some changes where we had some immune cells coming around the outside of the islet. And that's something, obviously, as you know, that's tends to be an early characteristic of perhaps something being,
insulitis.
Changing the immune system so that the islets are now recognised by the immune system as being something other than self. And so that piqued me instantly. I thought, oh, wait a minute, that's something I did not expect.
Is this in humans or mice?
No, so at the moment, that data was in mice that I was looking at. And so we took a step back at that point and said, okay, well, what if we inhibit advanced glycation in mice and see if that can stop it in models of type 1? So certainly we've published that. So that's in diabetes. Some time ago we published that if you inhibit that process, you can, slow down things. But of course, these models are models and they come with the limitations of models. And so we actually looked in some patients very early on in their first 2 years of life to see whether there was any difference in the advanced glycation end products in the circulation. And we weren't alone doing that. There was another group in the UK who looked at a very large school cohort and looked at the association of islet autoantibodies and these AGEs as well. And both of us showed quite clearly that they were elevated in those young people who were then progressed to get type 1 diabetes. And so the cohort in the UK was a really extensive cohort, and they actually showed it was a very good additive risk predictor on top of islet autoantibodies. So they had quite a good kind of risk prediction model in their cohort. And so at that point I thought, okay, well, I can't go any further from a commercial perspective with this age inhibitor because it died because of the global financial crisis. So I thought, how am I going to tackle this from another, way? And so that was where I thought, okay, what about RAGE, the receptor that these AGs might be interacting with? And so I started to look a little bit more at that, and certainly I showed that there was differences in that, and we did some human studies first to see whether RAGE polymorphisms in the gene changed susceptibility. Most of the work I've done there is in Finland because they have, as you know, they have the highest incidence in the world. I had a collaborator there that I knew from diabetes complications, and so I worked with him because he had access to very large genetic cohorts from Finland, including patients with type 1 diabetes, and so I started there. And so we produced a couple of papers with them looking at that, and certainly we could show not only that there was an association with the gene, but there was also that
with HLA subtypes, you mean?
Well, no. So basically what we did, so RAGE actually lives in the MHC region. So it sits smack in the middle of the risk HLA region. Mm-hmm. And so we obviously had to control for that as well because, as you know, it can just come along for the ride. It can be a spectator and nothing to do with the thing. Right. And so we also did some general populations, but we also did some where that everyone was matched for their HLA risk, and we could still show a very strong association. Wow. I mean, that still doesn't prove anything, but it at least gave us some confidence that we had something perhaps there at that site. And we also knew that certain polymorphisms, so there were two that characterised increased risk, and there was one that was protective, really protective. And
this is of RAGE. This is a polymorphism of RAGE.
HR is the receptor, so of RAGE.
Okay.
And what they did was in part a functional change in the amount of soluble RAGE in the circulation. So the patients had particular levels of soluble RAGE before in this risk period of autoimmunity development. Okay. So that again matched that AGEs were higher, soluble RAGE was down, something was going on with this system before the onset. And so therefore we thought, well, what about if we just give soluble RAGE and just normalise that? Let's see if we can stabilise the autoimmunity just by changing it in that period. And so that's actually what the preprint is about. We just tried to flatline the soluble RAGE in that period back to roughly what it would be normally, and to see if that would change the onset. So we literally gave the soluble RAGE for 2 weeks only in that autoimmune period, and then we stopped giving it.
Right.
And then we watched the development of diabetes. And so that's actually what's in that paper. It's that, and it's also the fact that we had a surprise finding. Yes, we had a surprise finding on the T cells. So that was something that we had never looked at before.
That was amazing.
I'm not an immunologist, but increasingly some of the work that I've been doing in some of my other areas is, so we actually have had already become quite good at flow cytometry and a few other tools that we needed from our kidney work. And that allowed us to look at this. And what it's also given us, which is not in that paper, is that grant. So that grant, I had an innovation grant from JDRF a few years ago that supported this work that you're now seeing come out. But what I now have that you may not know is I actually have another grant with Kevin Herald. So Kevin Herald's from Yale.
Yeah,
He's a
I
very
know
prominent,
him. Yeah,
So,
I know
Kevin
him.
Herald,
I know him well.
yeah.
He's a wonderful person.
So he obviously has had some interest in RAGE a long time ago. He obviously is a very prominent figure in type 1 diabetes.
Yeah.
But because of those two things, I went and spoke to him because I thought he understands RAGE. He's an immunologist, but he's also in TrialNet. And because at that stage we were toying with the idea of whether this biologic RAGE that we had, was something we could continue from a commercial perspective and what that would look like, or was there another approach that we could make? And so you'll see that the grant that he and I have got, so we've had some pretty serious conversations about that and also with JDRF. And so we've made a decision that we would go with testing RAGE antagonists. Now, there's one that he has used before and had a bit of playing with, and so we're working on that. But there is actually another one. I can't say too much about it because it's in confidence.
Right.
But I think the grant, I think if you look on the JDRF site, there may be some information about that, or I could certainly send you the non-confidential.
Okay, yeah, I'll take a look at it. But you have two different antagonists right now.
So there's two steps to that grant. So what JDRF have asked us to do in the first instance is bring up this second compound, and the reason for that is because the first compound doesn't have as easy commercial road because it's already in phase 3, another indication. It had failed that indication except in the patients with diabetes.
Wow.
So it did show a signal with them, but it's got a difficult pathway because it doesn't have a patent life. And so the reality of the situation is if you want a big pharmaceutical company to partner and continue, I mean, JDRF are considering it anyway and will find a path for it. There's no question if it works.
Yeah.
However, there is a second compound that if it works just as well, and the safety data is in place, which at this stage I don't know from the company, to be honest. We're waiting for that. They may actually already have that. Then that's the next step. So basically we're doing 2 years on this project looking at specific effects on models where we think it will give us an indication of whether it's going to work in humans. That's really what we're at. So he's doing humanized models, and I'm doing particular immunological models where we're looking at incidence and we're removing T regulatory cells in the same way as we did in that preprint to see if it's working in the same way as the soluble RAGE, because it may not be.
Yeah.
The reason we went down this path first, because we did produce both to JDRF, but obviously it's a finite pot of money.
Right.
And we thought these are both orally available. We thought that was obviously an easier path. There was a lot of extra thing, less expensive. So we did make that choice based on a few discussions, and we made some hard decisions there. It doesn't mean the soluble RAGE has been dropped. We're certainly bringing that along because it may actually ultimately, I don't know, we may be able to.
Wait, can I just clarify something? So in the very first compound, you said that's not patentable because it's already in phase 3.
It is patentable as it was, but I don't think the patent for type 1. I don't think you could patent it because
not
I think
patentable for type 1
iteration.
I got it.
In its first iteration, I think it had one of those patents that said, "I will fix everything," kind of thing. And so because of that, I don't think there's any life in that. I would suggest, certainly from our TTO's perspective, that would be what we would be saying. Certainly that's,
So the soluble one, though, so when will those 2 years be over with testing your models?
So in the 2 years, so we've started this grant. Mm-hmm. So it finishes about September. I mean, give or take what's happening in the world at the moment. We're hoping that that will still be online and ready.
Yeah.
But we're certainly on track. So we've got the compounds from the pharmaceutical company. All the agreements are in place. We've got the models started. So we're definitely going. And Kevin and I obviously have the money. I'm the PI, but Kevin is the CI on there. It's not because I'm the PI. It just made it easier for him. He's a busy man.
Yeah.
He didn't have to do most of the paperwork, is the truth.
That's great. It's great. Whatever moves the ball forward, believe me.
Absolutely. And I said to him, look, I'm happy to take the administrative burden because I know that you'll still have to do stuff at Yale anyway, without question. But if I take this, can we do this?
Right.
And so he said, absolutely. And so, so we got that money. But the vision is that our we actually had put in a 5-year grant. because that was the RFA that came out. So they asked us to do these 2 years, and then what they want to see is, okay, well, what's our plan? Because we originally had said we'd take the other molecule straight into clinical trials and not do this work because we didn't appreciate that the pharmaceutical company had a second compound.
Yeah.
So that was the original plan.
Well, the best laid plans, but it could be a silver lining.
Look, absolutely. I completely agree. And that's why we agreed and said, oh, absolutely, we will test it and see if it's the same efficacy. So that's what we're doing at the moment. We're trying to understand which one's better, which one where we should put the eggs in the basket of. And then, and in 2 years we should have a pretty good idea.
So for that trial, is that on Clinicaltrialsgov right now?
It's not, because we're trying to keep that quiet. I think it's been discussed. I don't know with respect to putting it out in the public domain.
Yeah.
It certainly was in our grant, but I don't know. I'll have to have a look and see what was published from the original grant, because what happened was we put the original grant in, and then JDRF came back to us and had a discussion and said, well, stop, we want you to do this because we know extra information that you don't, because we've had interactions with this company before. Let's have a conversation about that, and then let's progress from there. So that's exactly what we did. And so that's what JDRF are doing much, much better, I have to say, is they're not just coming back and going, no, we're not going to fund you. They're coming back and going, wait a minute, we know something else that you don't.
Which is
Let's
better.
Change. Absolutely. Yeah. Because that enables us to then take on those toolkits, that angle, whatever it is.
Good.
To
Sanjoy and Jessica are evolving. That's wonderful.
Oh, no, absolutely. And Sanjoy I've been dealing with because we're doing a clinical trial in type 1 and diabetic kidney disease that we're just about to put on clinical trials. So that's something separate to what I'm doing at the moment. But in this prevention space, I guess, also thinking about the regulatory authorities, you have to do one in newly diagnosed patients originally. And so that's often a safety study. What we're not looking for there is efficacy. We try and set up endpoints to have a little test and see, but that's not actually the reason for the trial. The reason is just to make sure that it's something that when these patients take it in the long term, it's safe. Now, we've got a bit of an advantage with one of the compounds because it's already been given to people with diabetes, so we know that it's safe already, but they would still like it because they're often younger people, so there's an extra layer of regulatory, which there should be.
What is the, let me ask you, so with the second compound, what is the age group that's targeted? Is it just young? We were saying two years old.
We assume that it might be available for whoever in terms of at the moment we're targeting it for prevention, but we don't know that this compound is more likely than the soluble RAGE to work at diagnosis. The soluble RAGE is more likely to prevent the autoimmunity. So probably a slight difference in their mechanism of action, I would suggest.
Yeah.
But I don't know that for sure, but that's what Kevin and I are speculating given the data we already have.
Okay. So I'm going to write this up and share it with you, and then you can approve it, obviously, right? Because this is research absolutely. To researcher. Only researchers, only students, only postdocs. It's a curated space. So it's not like, people off the street are going to be reading this. I just wanted to just give you that caveat. But also, let's have like a thought experiment. What would be a thought experiment? We could say, like, if you gave this new compound, thinking outside the box, to someone who's already had this, say, for 5-10 years, maybe, you've got the memory cells and all that. So maybe you're not going to turn everything off. But what might happen?
So one of the things that I guess is misconstrued a bit is that everybody's looking for a prevention therapy.
Yeah. Or
everyone's looking for something that absolutely to give a diagnosis to stop everything from happening. But the truth is that a lot of the devastation comes after that. There's so many other things to deal with once you have the disease.
Mm-hmm.
So the idea is that anything that prolongs the life of the beta cells, changes the immune function, reduces their risk for complications, which we also believe this therapy may have efficacy for. So we will, because I'm in complications, I've already had a conversation about that too, because that any year that you give a person with better beta cell function reduces their risk for complications phenomenally.
Yeah, and decreased glycation of, not just the A1Cs, which are like the, canary in the coal mine, the rest of the whole vasculature and everything. So,
look, I'm interested. I had originally done a lot of work with sRAGE and that area in complications and things, and it basically switches on that same inflammatory pathway that then leads to fibrosis of these organs. It's
right.
There's no question that it would probably have efficacy, and the vision is that we would give this for good, probably this agent. It's not something. It's a little bit different to and there's lots of reasons why we picked it.
Yeah. One question I have, I'm a big fan of Doug Melton's, and I'm in conversation with him regularly. But some of there's a lot of talk around the implant. I'm sure you saw that he sold Sema to Vertex, and now that's going to move into the clinical trial space. But the one of the issues I think that's been plaguing them, him, Dan Anderson, even Biocite, is fibrosis. Quickly comes and chokes off basically the implant. So, do you have any commentary about what things like soluble RAGE or anything in the genre how that could act on these implants?
I think there's a lot of work in kidney disease on that whole antifibrotic side. So it's something that I see a fair bit, because a lot of patients come in with type 2 diabetes and their kidneys are already shot. Like there's no going back. So you almost have to look at something for going backwards, if you like.
Yep.
So an antifibrotic agent or a fibrosis regressor, if you like. And so I think there's a lot of work going on in that space, and I think that will potentially be translatable to these types of things. From another space, the implant that you're talking about, is it one of the skin implants that you're suggesting, or is it?
Yeah, this is sub-Q. They're going to do sub-Q. Well, some people are doing sub-Q, and some people are doing it under the omentum. That Biocite's with the omentum.
Yep. So I don't know if you've seen some Australian work. There's some fantastic work that I think has now been released because the patent is in place from Adelaide. And so they're doing this stuff, I think they call it skinsulin. And basically it is how they discovered this way to look at the islets was actually, we had some really serious burn cases from the Bali bombing in Australia.
Oh, wow.
And the problem was that they couldn't actually skin graft because there was no skin left. So what they had to do was build a matrix to actually grow the person's skin cells into, and then put that in and then revascularize, and so grow the implant that way.
Okay.
What they've now done is adapted that to islets. So they've done all of the early preclinical work is done, and they're actually in clinical testing now, I believe. And they can certainly, the beauty of this one is that you can give topical immunosuppression. So you can basically use a cream on the spot to immunosuppress it. So that has been a huge problem, as you can imagine, giving a global immunosuppressant to someone who is already their immune system is already questionable. So,
and what is who's that group out of Adelaide?
So I think it's led by, I'm pretty sure it's led by Toby Coates. I'm just trying to think what his full name is. So he's actually a nephrologist, which is again why I know the work pretty well, because he's come from a transplantation space. Yeah. So he does a lot of kidney, pancreas transplants, but they also have islets available to do research with, and that's why it sort of has, I guess, given the opportunity for this. And the burns unit were also in his hospital, so they teamed up and thought about that. So
serendipity.
Yeah, exactly. Look, absolutely. So, there's no question that those opportunities, when they come up, need to be funded. So when that's presented to a funding body, the funding body needs to say, this is an opportunity that these things have suddenly come together, and this is a really and at first it was a very out-of-the-box idea, but they were very lucky. The commercial company who set up the implants realised that it was worth pursuing, so they did get some money originally to set it up in that manner, so that actually helped them. Because the Australian government has put a fair bit of money into those very early things that look like probably from a commercial perspective. Now, that's a very selfish thing from a commercial perspective, but in terms of money for the government. But what it does is gives at least an opportunity to these serendipitous
Yeah.
Things that seem a bit harebrained.
They do seem, yes.
A bit harebrained.
But I mean, it just gives it a little traction and see what happens. As part of our site, we actually have something called, this is going to be called Tinder, basically, where, just a play on the word, but we're going to have a place where a scientist can, kind of do a quick elevator pitch with 3 slides. This is what I'm doing. Who wants to collaborate? And the other person can join them, and then eventually we'll have funding monies there that they can, possibly write a grant together and then get funded there. So that's another part of our site. So anyway, okay. So,
yeah, and I think also encouraging people to, and JDRF has been pretty good at this, but encouraging people to overcome that's a competitor, because really on paper, Kevin Harold and I were in competition, if you like, in this space. And I just said, that's ridiculous. We're both on the same pathway. He knows a lot more about the immunology, but I know a lot more about the pathway of sRAGE. It seems senseless that we're not.
I totally agree. And I went to San Francisco last week before this whole thing blew up, or a week and a half ago, and I met with Jeff Bluestone. I met with Matthias, Hareth. I met with
Von Hareth,
and Von Hareth and Matthias Herzog. And so I met with all those guys, and this is what I've been doing is kind of getting the feedback. And surprisingly, despite some of the big egos that are in the space, a lot of people have been saying that this is the only way forward. So just what you said.
I agree. And Von Hareth has also been fantastic for us, because I also ran into him at a meeting, and again, a complications meeting that I saw him, but I said to him, I need to talk to you. I want to talk about soluble RAGE, and the data that's the second site in our paper was facilitated by him. So they replicated our data. So I gave them our compound. They analysed it, made sure it was up to their GMP standards, and then they actually repeated our studies at a couple of doses. So that was also reassuring for me. Totally different site, totally different set of hands, same data. Right, okay, this is not only safe, but we're getting the same result. In fact, we got an even better result in theirs because they had a higher degree of diabetes in their model. So
that's great. That's so fantastic.
So he was fantastically facilitative, and he also gave us a little bit of guidance about what needed to happen next for a next level investment. He said, look, type 1 diabetes is a smaller disease pot from a market perspective for companies, and this is the type of thing that these are the types of things that you need to be thinking about, because I'm not a commercial expert. I have absolutely no clue, but trying to understand the steps is actually very important, because if you don't know which is the most viable or useful way forward, you'll never get anything there. And I guess I've been lucky in my complication space is there too. So I'm learning from both sides. JDRF Australia have been supporting my complications journey there. And now JDRF International, I've just had an EOI approved for a clinical trial in complications. So quite different target, but a similar process of understanding that what steps need to be had and what is feasible and safe. So I think that's actually a really good skill set, and not a lot of us have that.
Right. So you're saying that JDRF internationally in Australia has been really a wonderful partner in educating scientists in the commercialization of their products.
Yes, I actually had a specific grant in Australia so JDRF Australia got, in addition to the money that they raised for the international pot, they also got some other money from the federal government for clinical trial development. And one of the things they spent some of that, a small proportion of money on, was to train people in this intermediate space. If you've got something that's got good safety data up to a certain point, how do you get it to the next bit?
Yes.
What are those steps? Who do you need to go and knock on doors to actually get that to happen?
Okay, that's really good. And they have you been involved at all with Helmsley?
Helmsley have been fantastic, because something that I didn't talk about before, and probably is relevant to your question before about the longitudinal effects of these therapies, is we also know that RAGE is very prominently expressed in the islets. Now, we've been doing some work with InPod, again, a wonderful JDRF initiative where we get access to human islets. I
have a call with Mark Atkinson next week.
Yes, so they're fantastic. So we've been working with, now I can't think of his name, Alberto Puglisi essentially they gave, and Helmsley Trust supported this work for us. Yeah. So we had a very early look. Again, I'm not an islet biologist, so we had some very good support and discussion with Alberto and NPOD, who are islet biologists, and I also spoke to people I knew around me who were. And what we did was we actually took a look in these young people and mostly younger people, so who had very generously donated their tissues, and we could actually see some very specific associations with RAGE and inflammatory pathways. That's not published data yet. We're just finishing it off. We're doing some work in human islets from our transplantation program in Australia to finish that off, but that's coming too. And certainly Kevin and I have discussed, we're going to look at that as part of our grant. It's not part of our funded component, but sometimes we've got the expertise to look at a few extra things, which is exactly what we're going to do. We're very interested in that longitudinal protection, because we certainly saw it in our preclinical models. We not only saw that it was safe with the soluble RAGE, but we saw that it was long-lasting, like right out through the model. They had lower, so even their risk for type 2 diabetes probably is dramatically reduced. Like it doesn't, all of the things are, so it's a longitudinal effect by reprogramming. And so what we would like to do with that soluble RAGE is move the goalposts around and give it at different times to see, do we get a different effect if we give it when the mouse is diagnosed, and then go back and, because that's obviously the step, okay, then how would we administer it to, because a biologic really has to be administered in a short, sharp blast because people develop antibodies to it. Mm-hmm. And so that was our rationale for that short intervention. Let's do it. Let's see if we can make any difference, because there's no point, because that's the window of opportunity for humans. We do know that. We know once you've got 2 autoantibodies, right, let's say
that's it. You're on your way. Yeah.
Exactly. So we think you're on your way, and that's probably the only time perhaps in prevention that they might allow you also to give a therapy, because that's such a high chance then that may even become the diagnosis point, so that people aren't forced into DKA and other complications.
Yeah. I mean, it's definitely been the whole thought process around it. It's almost like, it's sort of an in and out of remission. Once you have the one antibody, you're going in and out of remission until you enter DKA, essentially. It's just, that's the first step.
Absolutely.
Have you been able to quantify the RAGE levels is that what you're doing now, is to quantify these levels as the progression continues?
So after diagnosis and into...
No, after, the first biomarker is detected.
Oh, so we've actually already done that. So we've published that in a Finnish population. But
that's the Finnish paper. Okay.
So there's actually another, so there's 4, I think there's 3 papers, one more recently, which is, I think that's in Diabetes Research Clinical Practice or something like that. That particular one was actually matched samples from a very, very large cohort, and it's not a huge number of people, but to get them matched like that, you actually needed to look at 16,000 people to get 163 because they're so perfectly matched. So what that is that's a group of people, and that's how we know that the soluble RAGE starts to go down at seroconversion, and you can see in that it's progressors versus non-progressors, and it's a longitudinal look at seroconversion, the exact moment of that. Because these people are screened from birth, we actually have the data of the exact time when they converted, and then over the next 5 years what happened, and we can definitely see that there is this U-shaped drop in the soluble RAGE as their autoimmunity worsens.
Who's the first author on one of the papers?
I think that may be Salonen, Salonen. I'm pretty sure that's...
I think I've seen that paper.
So Kerstin was a PhD student that Michaela and I were both helping, and so she did 3 studies with us. So she did the 2 in the larger Finnish cohorts, which are both in diabetes care, but then there's a third one actually, which is interesting, probably the most relevant one for us in terms of plotting the time frames for that autoimmunity development, because we know at diagnosis the RAGE actually goes up, and the reason for that is because RAGE is a molecule. The reason that it's located in the MHC region is because it plays a role in dendritic cell antigen presentation of antigens, the mobilization of macrophages. So it's an inflammatory thing. So once once you've hit that point and inflammation just really escalates, you've got a cytokine storm and all those things that are happening at that time. RAGE is very high.